Thanks, Imre, and thanks also for organizing this event, which I'm really curious to see where it leads to, especially about the hackathon at the end. Thank you all for coming. So Imre has asked me to begin by giving a broad introduction to animal senses, and it's going to be really broad, and I hope I'm not just telling you things that you already know, but I'm going to be talking about that in the beginning, and then I'm going to be focusing on the research we're currently doing at Newcastle University, looking at 3D vision in the praying mantis. So that I'll get to in more detail. Of course, the idea behind giving you this broad introduction is perhaps to just give you points of reference from which you could explore further and telling you where to look if you like. And also the part of the idea, at least the way I've structured the introduction, is to try and give you an idea of what the questions which sensory biologists might be looking at in animal biology and why we do the things that we do. So this is, by the way, a picture of a robber fly, which we will be hearing a little bit more about from Paloma when she gives her talk, not from my talk. So I'm going to start with perhaps a very obvious, a very simple, silly question called about why have senses at all. And of course, if you ask a silly question, you might well get a silly answer. And a simple answer perhaps is to gain information about the world. So you would not be able to know anything about the world without senses. But if you go one step further, and especially when you're thinking about animal senses, the bigger question is, what do you use this sensory information for? And animals use sensory information for a whole host of different things. I've listed a bunch of them here, but you can think of other ones. So you could think of animals using sensory information for navigation, like turtles, which use magnetism to manage their huge migration. Birds do the same thing. Navigation can be important for insects that are central place foragers. So you have a hive from which you go out and have to return. So you use your senses to manage to navigate the environment. You also might use your senses to find mates, to assess the quality of mates, like in deer, which use acoustic calls to assess the quality of their mates, but also frogs and crickets, which also do the same thing. Going one step further from just finding mates, you might also want to use sensory information for communication. Prairie dogs use alarm calls to communicate, but you could think of olfaction in other rodents also playing a huge role in communication, and there are several different examples of this. Sensory information you could also use to find food. Of course, I've put up a bee here, but you could also think of finding food in terms of finding a food that doesn't want to be found, and that relates to the other point which I made, which is avoiding predators. What's important across all of these things, I think, is what the sensory information is being used for would determine how it's going to be processed as well. So it is important to take into consideration what the function of your sensory processing is to interpret how a particular animal might be using the sensory information. So if you like, function might also have a lot to do in interpreting the form of sensory processing. And when you think of all these different categories, you can also think of it, these categories fit very well including in animals, including humans. So humans also use sensory information for very similar things, including navigation, finding mates, communication, finding food, and perhaps even avoiding predators occasionally. So the point I'm making here is also that when you compare humans to other animals, or if you look at sensory information processing across different animals, we find several common problems of fulfilling these same functions. But the solutions which different animals might evolve might be similar or different depending on what the animal is. And why it's interesting to compare, say, humans with other animals, or even amongst other animals, to compare sensory processing in these different animals, is precisely to discover the different solutions that might arise. And given Imre's introduction, that would also be perhaps part of the interest for people who are involved in the hack the senses idea. But I'll come back to this theme a little bit later when I talk about the Mantis project. So the next question is, of course, what kind of senses do animals have? So what kind of solutions might we actually find in animals? And again, if we start from this notion of what senses humans have and look for these in animals, we do find evidence for several things that we find in humans. So you might have vision, sight, sound, vision, hearing, smell, touch. All of these things would be present in animals, but you do find them in very different forms. So you might have compound eyes or avian eyes, which are quite different from each other as well as from human eyes, but you might also have different kinds of ears. That's a bat ear. We'll be hearing more about bats later, or a lizard's ear, which can function in a way to, in the same modality, to process the same kinds of input, but process it very differently, and it leads to different consequences as well. So it's worth considering how each of these sensory modalities are processed. So for example, you could think of smell as just some way of processing chemicals, which an insect's antenna, a moth's antenna might do as well. Taste being processed by a butterfly's proboscis would be, I don't know, comparable or not comparable to how we might think of taste, and this is part of what we should perhaps start thinking about when we're comparing different kind of sensory processing. Touch is, of course, sensitivity to vibration or touch could manifest in different ways in the animal kingdom, from hairs on a spider's leg to perhaps a gecko's pads, which are also used for adhesion on surfaces. So you could get a whole wide variety of ways in which the same sensory capabilities or the same sensory modalities are sensed in very different animals. So even if you restrict yourself to the senses which we're already used to thinking about, you could find very different examples of these in the animal kingdom. And in a way, you could argue that they are different senses for these animals as well. They're just using the same modalities. Of course, the interesting thing about animals is also you can find diversity beyond what we see in humans. And again, this can be in many ways. You could find within the same modality like sight, they're sensitive to parts of the spectrum which we can't perceive. So you could have birds and bees that are sensitive to UV and might see flowers very differently from what we see them as. You could also have animals that are sensitive to ultrasound, again bats and dolphins that are now perceiving worlds which we're not aware of even within the same modality. Other animals are, of course, sensitive in modalities that we can't really perceive, including electricity, like this electric eel, weakly electric fish, that use electricity to communicate and to hunt for prey, which of course we don't do. And there's also really interesting stuff coming out now of bees being sensitive to electrosensation as well. So there are different animals that also are sensitive to electricity. Several animals are also sensitive to magnetism in many ways. So you find, of course, birds use magnetism to navigate on their migratory paths. Even roundworms have recently been shown to be sensitive to magnetism as well. So there are all these different worlds of perception which we don't have access to in a way. So given this, I mean, I of course find this variety of sensory capabilities quite fascinating, but I guess for several people, you might be well justified of asking the question of why study animal senses at all. Hopefully part of the inspiration of this event means that you already have an answer to this question. I've proposed three possible answers, three things which might motivate us to study animal senses. The first one I think of is just to understand the animals themselves. This, of course, has implications for us as well because we interact with animals in so many different ways, including pollination or pest control if we just restrict ourselves to insects. But you can think of many ways in which it's useful for us to know how animals sense the world. The other thing is something which I briefly touched upon earlier, is it's an interesting question to ask how sensory solutions evolve. So given the same common problems which I was referring to earlier, do we come up with the same sensory solutions or very different sensory solutions? Can nervous systems solve the same problems only in one way, or do you get different ways of solving the same solution from different nervous systems in different animals? And if you do have different solutions, then the question is, can we also use the solutions that other animals have? And so I've broadly categorized that as technological solutions with different kinds of robots or computer algorithms that could tap into the algorithms that already exist in nature. And so with these three broad ideas, I'm going to stop my introduction to animal senses, but show you how perhaps we've used these three ideas as inspiration for work we're currently doing at Newcastle University. And that is in a project looking at stereo vision in the praying mantis. So this is Newcastle University, which is, well, different from University of Newcastle, which is in Australia, I guess. So we always have this problem, so we have to keep doing this. And this is part of a project called Man, Mantis and Machine, as Imre pointed out. It's funded by the Leverhulme Trust on a grant to Professor Jenny Reid. The work I'll be presenting has been helped by several other people whose names are here. we have a computation engineer on the project, Guy Taravane, and a neurophysiologist on the project, Ronnie Rosner. I do the behavioral work on the project. And the idea behind this project is similar to those three themes I presented. We're looking for the mechanisms of stereo vision in the praying mantis, trying to see how the mechanisms differ from what we already know about stereo mechanisms in primates, in humans. And the idea is if we can adapt those solutions, if there are new solutions found in insects, we would ideally like to come up with computer algorithms inspired by insects at the very end of this, hopefully even adapt them into robots. Depends on what guides can do at the very end of the project. So that's the inspiration behind the project. I'm going to start by giving a very brief summary or idea of what stereo vision is for those who might need it. Stereo vision is the ability to compute depth by comparing the two different views of our two eyes. So each eye has a slightly different view of the world, and the difference between these two views, which I'm going to call disparity for this talk, you might hear the term later. You can use this difference between the two views to calculate the depth of an object. So objects that are closer will have more of a shift between the two eyes. Objects that are further away will have less of a shift. So you can tell how near or far something is just using the difference in these two eyes. So this is something these old stereo cards used to make use of, or if you were as a kid using those, I don't know what they were called actually, those stereograms, you could see slightly different views of each eye. So even now if you try to fuse these two, you should be able to get some kind of depth percept, so, which is what our brain does when we're trying to compute 3D information. So 3D vision of this kind has been seen in several different animals. So again, it's a good system to look at different solutions to the same problem as I was talking about. It's been seen in other primates, in cats, in toads and horses, and also in owls and falcons. Amongst these, if we discount toads at least, but the kinds of mechanisms have almost definitely evolved independently in birds and in mammals. So now you have two different evolutions of the same phenomenon, if you like. And it was also discovered in a third independent lineage, which is the praying mantis. So the praying mantis is the only invertebrate from which we currently know 3D vision. And it's a really interesting system to study, partly because it's so evolutionarily removed from primates. So if there's convergent evolution, you might expect very different solutions, possibly. It's a visual predator. It has front-facing eyes with a huge degree of binocular overlap, so it's really good for judging, comparing what the views of the two eyes are. They have a particular, well, often I should say, have a hunting strategy where they stay in wait, stationary, until prey passes by at a specific distance, and then they reach out and grab it only if it's the particular distance. So you can imagine that judging depth becomes really important for mantises then. And that's why it was, I think in the 70s and then later more definitively in the 80s, were the first experiments that showed that praying mantises had stereo vision. Those are pretty cool experiments, but I'm not going to get into them right now. We could have a chat later about that. But what we wanted to investigate further is what the mechanisms behind this were. How is it actually computing depth? Is it similar to us? Is it different? Is it doing something completely new? And it's been very difficult trying to find that out using the kinds of setups which were used previously. So we had to come up with something new, and after a lot of trying out different things, the solution we hit upon was to construct a 3D insect cinema. So what you see here is a praying mantis with two different colored glasses on each eye. So one has a blue glass, that's that one, and one has a green filter on top of it. We can, with mantises wearing these two filters, we can show them colored stimuli on a computer screen so that one eye sees the green stimuli and the other eye sees the blue stimulus. So each eye can see two different stimuli, and you can use that to manipulate the 3D perception of the mantis. They like hanging upside down, that's why it's there. Just for the videos as well, you'll see that they do hang upside down. So what you can do if you want to manipulate the 3D images is you can either present them in three different ways. So in the first way, the lines of sight cross. So each stimulus, each eye sees a stimulus that is on the opposite side to that eye. So this eye sees the blue stimulus and that eye sees the green stimulus, and the lines of sight cross. And if, for us, when we had 3D perception, if the lines of sight cross, we perceive the target to be where the lines of sight cross, so over here. So even if the target is 10 centimeters away, or as we presented to the Mantis, you can manipulate it so that it perceives the target to be only 2.5 centimeters away. If the lines of sight coincide on the screen, then you see the target on the screen, so it's a 2D stimulus. So that's a normal 2D stimulus. If the lines of sight meet behind the screen, then you see the object further away, or if they don't meet at all, you don't get any illusion of 3D. So those are the three conditions, if you like, which I've called 3D near, where you do expect the Mantis to strike if it has 3D, 2D, where it shouldn't strike because now the target is too far away, and 3D far, where there is no real coherent target, so it shouldn't really strike. So this is where we expect strikes and not in the other two conditions. And what we were looking for is, can we actually do this? Do mantises actually see 3D illusions? So this is the same kind of setup which is used in the cinema. It's just that it used to be with color and now it uses polarized light. So that's the difference. So this is the stimulus we used. We use green and blue for the mantises, but I've used red and blue here so that you can see it if you put on your glasses now. And I'm just going to play it now, and you should see the stimulus, well, depending on how you're wearing the glasses, either in front of the screen or behind the screen. So if you reverse the glasses, you should see the opposite, and it should continue. So one of them is in front of the screen, and then reverse it, and you see it behind the screen. So this is stimulus we presented to the Mantis, and let's see what they did. So this is a normal video, not a 3D video. This is for the 3D far condition, but it's a similar response to the 2D condition. And that's the stimulus swirling in. So it's green and blue for the Mantis, and the Mantis does nothing, which is good. That's what we predicted. And then if we show this same thing with the 3D near condition, now we expect the Mantis to actually respond as if there was a nearby target. And we tried this out on the mantises, and yes, so that's what they're doing. So that's what they do typically to prey. They reach out and strike at the prey, and they did this with the 3D near condition. So if we do this over several mantises and look at the number of strikes per trial, so the mean number of strikes for each mantis, if the screen is 10 centimeters away, they really shouldn't strike. And you do find that for the 2D condition and for the 3D far condition, they don't strike. But for the 3D near condition, they do strike. And they only strike for the 3D near condition. So basically, this was really good news for us, because it showed, of course, definitively that praying mantises do use 3D vision. But also it showed us that we can use our display setup, our 3D virtual setup, to show mantises 3D stimuli and more complicated stimuli with which we can now start exploring the mechanisms of 3D in praying mantises. One of the first questions we wanted to look at in praying mantises using that setup then was to compare it to what we know from primates and from owls. And what we know from primates and owls is that 3D vision helps them break camouflage. In fact, it's been hypothesized that that's the reason it has evolved in these two animals. So again, going back to the function of sensory processing, this is partly how we get at that, in terms of if we think of 3D vision as having evolved to break camouflage, we can now ask whether mantises also can break camouflage, and that's the reason 3D vision evolved in the mantis. And to show you what I mean, if you don't have a camouflage object, just a red triangle, you see it, this is in the right eye, this is in the left eye, and then using 3D vision, you see it popping out of the screen. But if something's camouflaged, when you see it with only one eye, either the right or the left eye, you don't see it popping out, and you can't see an object at all. But when you do use both eyes and you see it using 3D vision, it pops out, and then you see the outline. So that's how stereo vision can actually help you break camouflage. And how this actually works in the primate and the owl brain is through cross correlation of the two images. So the neural mechanisms in the brain are sensitive to matches between these two images, and it actually matches the entire visual scene from the left eye and the right eye. And when these matches occur, you know that there is basically that's when the object pops out in stereo vision. So we were really interested in seeing whether praying mantises also do this cross correlation and manage to break camouflage. So to do this, we used a camouflage target stimulus, of course, and you had both the target and the background. So the camouflage, which camouflages the target, consisted of really small dots. But we did it initially with a correlated condition. So the black dots in the right eye and the matched the black dots in the left eye. White matched the white in both eyes. So basically, for every dot over here, you have a dot in the other eye in the same position and with the same color. Yeah. So this is an example of the stimulus, which you should be. Yeah, sorry, I should have told you. I don't know if it goes on loop, so I might have to play it again if I can, or I might just go back and play it. Yeah. So in these stimuli, we can perceive depth. So we see it as either in front or behind the screen, and we were curious whether the mantis can do this as well. And I just wanted to point out that on an individual frame of each of these stimuli, you can't see the target at all. So the target is given away only by the 3D cues, the matches between the two eyes, and by the motion. So these are the two cues which it's using. And we wanted to see if the mantises can do that. And what we found was the mantises did manage to do that. So they were managing to break the camouflage, and they were striking more for the 3D near condition and almost not at all for the 2D and the 3D far conditions. But there were two different cues they could be using. They could be somehow combining the motion cues, which were available to both eyes, or the static position cues of the dots. And we were curious to see which one of these the mantises were using. So the first thing we did was to try an uncorrelated stimuli. So now the dots don't match up at all. And the primate mechanisms and the owl mechanisms would not be able to see depth in these stimuli because the match breaks down. And we wanted to see if mantises would also have a similar kind of response. Here's what those stimuli look like. If you put on your glasses. So I don't think I've ever looked at these through glasses, so I don't know if you perceive this as anything at all in front of the screen, behind the screen. I don't know. What usually happens, of course, if there's an ambiguous stimuli is humans, in psychophysics, we tend to say that it's in front of the screen, but we do that regardless of which way the glasses are. So basically, we're not perceiving depth. We have a bias towards saying things are in front of the screen. So we're really bad at perceiving depth. When we ran these experiments with the praying mantises, we found that they still are fairly good at perceiving depth. So they do respond still more to the 3D near condition than the 3D far condition. So essentially, this disruption of the matches does not disrupt the praying mantis's response at all. So possibly what they're using then is the motion cues. To test this, what we did was try to remove the motion cues by having the background move at the same time as the target. So it's camouflaging the motion cues of the target. And this is what that stimulus looked like, if you're not tired of putting the glasses on. The black dots are the background. They're just moving. And you have a focal area that moves over the dots. So there's no clearly defined target boundary. The target and the background consist of the same dots. The only difference is that the focal area has a shift introduced by disparity between the right and the left eyes, but the other areas don't. And we were curious to see now if this disrupts the mantis' ability to perceive depth. And what we found was that it actually does. So mantises now start striking even at the 3D far condition, and essentially there's no difference between the 3D near and the 3D far condition. So disrupting the motion boundary of the stimulus removes or impairs the mantis' ability to actually see depth. So what we think is happening is that target motion contributes to depth judgments in the mantises, and possibly they're actually correlating the motion cues in both eyes rather than the static positions or the of the dots in both eyes. So just to summarize, we've used our 3D cinema setup to actually show that we can use it to give 3D illusions to the mantises because of their 3D vision. And they use this they can use their 3D vision to spot camouflage targets that are camouflaged with everything except motion cues and disparity cues. But they use they possibly use motion cues, and therefore their mechanisms might be something we don't know at all from primates. And so, I mean, that's a really interesting result for us to find, because now we can go ahead and find out what those mechanisms are and come up with something potentially quite different in mantises compared to primates. So I'm going to stop there, and I think questions are at the very end. So if you have any questions, I'll take them later. Thank you.